Yeah, I want to say thanks to JGI for making a lot of my career possible. So I started collaborating back when I was a brand new assistant professor, and it was the opportunity that I got through CSPs that really generated some of that first data that led to my first publication. So, and this is the first time I got to speak at a user meeting, so thank you for inviting me. I'm excited to tell you about something that's kind of a side project in the lab right now, but something that I'm really excited about that has come out of some of the larger CSPs that we've been working on in the past 5 or 6 years. I wanted to take this opportunity to remind you that yesterday was World Water Day, so if you missed it, you can still celebrate it and drink a glass of clean water and thank engineers for making it clean for you. And so we are going to be talking about water, and specifically fresh water, which is, believe it or not, in most ecosystem frameworks thought of as part of the terrestrial ecosystem. I mean, we sort of get lumped in with soil and other kinds of terrestrial habitats. It's considered as a distinct compartment for marine systems. And we do have data now that shows us that the freshwater pipe, if you will, is much more like a reactor in terms of carbon moving through the landscape. So carbon is fixed by plants on land. It then makes its way into the soil, and many of you study what happens to it there. But it eventually ends up in a water body, much of it. And the amount that ends up in a lake or a river or an estuary determines then how much ends up going into the atmosphere versus being buried versus going on to the ocean. And the amount that is captured in those freshwater ecosystems and either respired as CO2, releases methane, which also is a big contributor for the global methane emissions, or it's buried. And then there's a certain amount that goes to the ocean. And this is my chance to get up on my pedestal and say that freshwater doesn't get enough recognition in these global scale carbon models, largely because we don't know the fluxes and the pools accurately enough. And so that's one of the things that I work on with some of the ecosystem scientists that I work with. And this is a more simple way to capture that, which is thinking about inland waters as a pipe, but a very active pipe in terms of the biogeochemistry that's happening as water moves from the land to the ocean. And of course, microbes drive all of this. I don't have to tell many of you why microbes are so important in ecosystems. This is my view of the world. I think of mostly bacteria in my ecosystems. Eukaryotes kind of terrify me, so we tend not to study them as much. But the bacteria and the archaea carry out all these interesting biogeochemistry reactions, and carbon being one that we're very interested in, but also nitrogen cycling, phosphorus cycling, sulfur cycling. All this is going on in these freshwater ecosystems, and they're all coupled, and they all influence these things that we care about, like carbon flow and emissions through the system. Now, I'm really fortunate to be studying all of this in the context of something called a long-term ecological research site. So we heard yesterday about the Harvard Forest. There's a number of these around the country funded by the National Science Foundation for long-term, so they're 7-year grants. And the North Temperate Lakes LTER has been there for more than 20 years. I think it's more like 27 years now. And it's an amazing resource because there's people there that have been studying the lakes for years and years, who have lots of ancillary environmental data. They study the fish, they study the plants, they study everything else but the microbes. And so I come along and say, hey, I'd like to study the microbes. And they say, great, just tag along. And so we have access to all their infrastructure, all their historical data. We have archives of samples going back to 1999, even for the microbes. So I just want to give a shout out to NSF and LTER because these are amazing sites that, if you have a chance to partner with, you should. And one of the main things that has come out of our LTER site, this is by John Magnuson, who's pretty famous for his work on ice cover on lakes. So this is a measure of ice cover on Lake Mendota, which I'm going to tell you more about. It's the lake in Madison. And it shows one data point from 1983. And it shows that ice cover was about 50-something days over the course of the winter, right? So in Wisconsin, it's a big deal. Ice on the lake supports fishing and sports. People go out and ride on kites across the lake. It's kind of weird if you don't experience frozen lakes, but it's a big part of our culture. And so if you take that one data point, you see one thing, you take a few more data points, and you see, wow, that first data point that I had was actually pretty low. It doesn't represent at all really sort of the normal state of the lake in a particular year. If you expand that out to several decades, you start to see strong interannual trends on multiple scales. So in this case, you can see El Nino events really influence the low points of ice cover duration. I should point out that if you go farther out on this, you start to see a downward trend, which is the climate signal, the long-term climate change signal. And if you care, this year was the tied for the second shortest ice cover duration. It was about 67 days. So we're tied for the one that was in about, I guess it was 1960 or 1970. So we do see that long-term trend. We wouldn't see the long-term trend if we didn't have this long-term time series. So that's the take-home message, is that you miss a lot if you don't follow stuff over time, over multiple time scales. So the story I'm going to tell you about today is about Lake Mendota. Lake Mendota is in Madison, Wisconsin. So if you've been to Madison before, you'll know it's like sort of in the lower central part of the state. Lake Mendota is eutrophic. So it has terrible cyanobacterial blooms in the summertime. It's embedded in a mixture of ag and urban landscapes. It has invasive species in it, like spiny water fleas and zebra mussels now. So there's lots of insults hitting this lake. So we've been studying it for a long time, trying to understand how all these climate, land use, and invasive species type changes are influencing all aspects of the lake. And my role is to study the microbes. And so we are very fortunate to have what I call a data smorgasbord. I work with a lot of Swedes. So I think of this as just sort of this pile of amazing stuff that we get to look through. And so we have partnered over the years with the Earth Microbiome Project, with Bigelow Laboratory out in Maine, to generate, we have now more than 12 years of 16S sequencing tags, and we keep adding to that as the years go on. We have 5 years and counting of shotgun metagenomes, which is mostly what I'm going to talk about today, combined with now going on about 200 single-cell genomes that come from this lake. And we also have data sets from other lakes, and so again, I'm just going to say thank you to JGI for being so generous, and to the reviewers of our CSPs for supporting us to generate this huge pile of data, which for those of you sifting through your own piles of data, you know the challenges of trying to get graduate students up to speed on getting those out the door as papers. So I am a very organism-centric person, so I'm going to have to tell you a little bit about the microbes that live in Lake Mendota. And so this is just a rank abundance curve of all the 16S reads from that time series that I mentioned that were generated by the Earth Microbiome Project. And to point out that the top 10 or 7 or so taxa are present pretty much in every single sample that we see. So if you think about that concept of the invisible present and what's the snapshot that you're looking at, and when do you know that you've sampled enough to be able to say something about the organisms in the lake, most of them, the really abundant ones, are there all the time. So that was encouraging. We do have this is another chance for me to be on a soapbox and do a shout out to the concept of coming up with your own controlled vocabulary for talking about organisms that don't have cultured representatives. So we have a really structured hierarchical naming system for them, and I'm not going to go into the details of that, but if you are working on an ecosystem where you're dealing with like all these cryptic names that somebody invented 5 years ago, 10 years ago, that you may want to think about totally restructuring that and getting your community to adopt it. So the organisms that I'm going to talk about mostly are ones that we have genome sequences for, of course. Actually, we have genome sequences from most of the most abundant organisms in Lake Mendota, either as single-cell genomes or metagenome assembled genomes, MAGs. I'm going to talk mostly about the top 2 most abundant clades. So those are the AC1A and the AC1B, and I'll tell you more about them in a moment. But you can see they are the ones that are the most abundant, at least by 16S sequencing measures. So this shows a time series of 3 clades within the AC1 Actinobacteria. So these organisms are essentially very streamlined streptomyces. So think about a 1.2 megabase pair streptomyces genome. And so they basically lost all the stuff that makes them look like a typical Actinobacteria, but they are the most abundant and most ubiquitous bacteria in fresh water. So they are in every lake that anyone's ever looked in. Fish, so fluorescent in situ hybridization shows that they can be up to 50% of the cells. So they're kind of like Pelagibacter in the sense that they're everywhere, right? And they're abundant. So my lab is really focused on trying to understand both the population ecology and the community ecology and the traits and the functions of these organisms, as well as all the other ones that they live with, which is no small task. But the story I'm going to tell you about today came about trying to figure out how do we actually even keep track of these organisms in the lake, and what can the metagenomes tell us about how we should do that. And so most of the time I'm going to be talking about what defines an ecological or coherent unit within these groups of organisms. And then we can talk about at the end, if we have time, whether or not it matters to spend all this time defining these units in terms of the things that we really care about, like CO2 emission from the lake, which, you know, we're a far cry from that when we're talking about just how do we even keep track of the organisms. So what we have been doing a lot of is something that was pioneered by early people doing metagenomics. You think back to the papers from the Global Ocean Survey. You think back to Kostas Konstantinidis' work, Paco Rodriguez Valera's work, where you take metagenomes and you recruit them against a reference genome. Now, we are fortunate to have this great collection of both SAGs and MAGs that we can then take individual metagenomes and compare them to the reference genome. So what you're looking at is a typical recruitment plot that you don't see as much anymore in papers, but was fashionable a few years ago. But you can see that the y-axis is percent identity, so that's the percent identity of each individual metagenomic read. The x-axis is the genome coordinate, although since it's not a complete genome, it's sort of contigs stitched together in an arbitrary order. But what you can see is that there's a big smear at around the middle of the plot here, which is about 85%, and then there's sort of a gap, and then there's a lot of density up here, sort of above 95% identity. So, and this gap has been noticed before by other researchers. You can see it better if you turn this plot on its side. So you now have the percent identity on the x-axis and the density of the reads that map at that percent identity on the y-axis. And so what you see is that big hump at 85% or so, and then sort of this discontinuity, we call it, and a big spike again here up by 100% identity. And so working with Rex Malmstrom, we started using this language talking about sequence discrete populations where this discontinuity creates a discrete cutoff so that if you only look at the reads, the metagenomic reads that are mapping above that point, that you can think about that as the population that that SAG represents in the lake. And so this is an example of one SAG that came from Lake Mendota with a metagenome from Lake Mendota mapped against it. Now you can do other things. Oh, I guess I wanted to say another shout out to JGI. So they published a paper a few years ago, this is not with me, but from Nikos's group, looking at genome-wide ANI as a metric for delineating species. And so this is kind of a complicated plot, but the bottom line is that if you look at the far right-hand corner of alignment fraction, which is on the y-axis, and average genome nucleotide identity on the x-axis. So those are the organisms that are all 99% of them are pairwise comparisons of named species, so things that we're pretty sure are the same species. You start to get out of this region up here and down into here, and these end up being many more of the pairs of genomes that are not the same species. So they propose this metric to say, you know, by whole genome comparison, are these two genomes from the same species? So it's a similar concept that we're using. Again, this was something that Kostas Konstantinidis proposed way back when he worked with Jim Teege, so it's not a new concept. But the data that we have now allows us to look at these populations from the wild in a way that we weren't able to before. So going back to this plot, so here's your sequence discrete population delineation. You can look at other reference genomes. So this is a reference genome from a different lake. This lake is in Maine. But the metagenome is still from Lake Mendota. And so you'll notice that what happened is that you lost those high number of recruiting reads. And you know, the obvious interpretation of this is that the population that was represented by this SAG is not in Lake Mendota. It's a different species, but probably a different genus even. And so this is not a good reference genome for studying the wild populations in Lake Mendota. So you can take another one from a lake in Germany, which is farther away. But actually, there are some reads that recruit at those high percent identity. So it's not quite as clean as you might expect it would be in terms of thinking about the distance between genomes and whether or not you can rely on a single-cell genome from one lake matching what you find in the metagenome from another ecosystem. But if you overlay many different SAGs and many different metagenomes, all those different combinations, you can see that usually if the lake is from somewhere nearby, that you get this peak above 95%. So we can talk about biogeography and what that means for dispersal, but we're not going to have time to do that today. But pointing out that we have this large collection of SAGs, we have all these different metagenomes, and we can think about sort of what the structure of that population at above 95% is at any particular time point and then across time. Right? And so I know some of you who do evolution are thinking, or think about evolution, are thinking, well, talk to me about SNPs, talk to me about metrics that we can look at to which genes are the ones that have the variation in them, all that. This is an aggregate metric that looks across the whole genome because in most of the populations there's so much variation that you can't really use, or it's difficult to apply traditional popgen metrics to it. So it's almost at a level of heterogeneity that falls in between thinking about what a different species is, the ecology way of thinking about it, and then looking at within population structure. So it's a different way of thinking about what a population is, and maybe something that not everybody's quite comfortable with yet. So we asked, what does this plot look like over time? So if we take this chunk above 95%, and obviously if you have two different genomes, you see differences, right? So we see differences in the breadth of this hump. And when you look across multiple genomes, all from Lake Mendota, and here's one from another lake in Wisconsin. But what does that look like over time in the same ecosystem? And so we have this fantastic time series of metagenomes. So what we did was look at that distribution over time. And I've now collapsed it so that each point is just the average of that distribution. So each point has the diameter being related to the relative abundance in the sample on that date. So each column is a sample. And the intensity of the color relates to the average ANI. So the average of that peak above 95%. Okay, so darker colors means it's more similar to the reference genome. And so the first thing that you should take from this is that if you use SAGs that are from a different lake, the average genome, the genome-wide ANI is lower than if you are using SAGs from the same lake. That should make us feel better because it's basically saying if I take an organism from its own ecosystem, that's a better reference than one from another ecosystem. But you can see some patterns in terms of the abundance of the organism through time, and maybe some variation in the intensity of that color, but probably don't want to spend a lot of time thinking about it because it's not really the right reference genomes. So then you look at this group down here, which you can see more evidence for variation in the abundance and in the intensity, or so the average genome identity. And this happens to be my favorite group of organisms, coincidentally, is the AC1B Actinobacteria, one of the most abundant groups in the lake. And I guess what I wanted to point out here is that you've got what is, if you squint, you can, I don't know if you can see it up there, hopefully, yeah, you can, they're darker up until a certain point. So they're dark up until a certain point, and then they get lighter. So what we saw is that in 2012, we have a shift in the population that you can see at the level of genome identity, still above 95%. So it's still, the reference genomes are still good references for whatever population was now there in 2012. It's just that they're a little bit more distant from the reference population phylogenetically. I should point out that these two groups that I'm tracking in the box here are part of the same sequence discrete population. So if you look at the two SAGs and compare their genome-wide identity, it's 98, 99%. And so phylogenetically, they're the two SAGs that are most closely related in our data set. So that's encouraging that they have the same recruitment pattern through time. You can also look at some of the other organisms on here. This J17 organism that's right below the box here is obviously capturing more of the total community of these AC1 bacteria because the circles are broader, but they're slightly less, the percent identity is lower than the reads mapping to these reference genomes, which means we probably don't have a reference genome for the most abundant population in the lake. We don't have it yet. We also don't have it in our mags. We've checked. So whatever this elusive population is that we're trying to see, we haven't quite been able to see it yet. But we see its relatives. You can also see interesting patterns where you see things just completely coming and going through time. So you can, this is what I wanted to say about that new strain arising in 2012, that you look at the statistics on it, it is significantly different in terms of the period from 2008 to 2011 versus 2012. Has the same number of variant sites, so it's not as if a certain section of the genome swept out or something or got accumulated more variation. It's very evenly distributed in the same way. So it is just another population that has now taken hold in Lake Mendota. So we're very interested to continue following this. Of course, we would love to be able to sequence from 2013 to the current day, so we're working on that. I just wanted to throw out another interesting observation from this, because I've been talking only about the LD12, or sorry, about the AC1 Actinobacteria. There's another group in Lake Mendota that's very abundant. It's called LD12. It is actually the freshwater sister clade to SAR11. So if you study, I don't know who in here studies marine systems, but if you know Pelagibacteria, you know that there's many clades of SAR11, and LD12 happens to be, sort of very close to the marine clades, but they're quite different. Obviously, they live in freshwater. We've done some work trying to compare the freshwater and the marine ones. They have a very different population structure from AC1. So if you look at their recruitment plots, you don't see the hump at 85%. You see everything kind of smashed up above 90%. So it's much more difficult to delineate the sequence discrete populations in this group. And this is the only one, really the only group that we found that looks like this. All the others that we've looked at look like AC1. So they do seem to be the outliers in terms of how their populations in the lake actually represent or the extent to which they are dispersed in their genome identity. So that's something interesting to think about. What about the lifestyle of these organisms makes them look like this as opposed to the others? Now, if you are interested in ecology, then you might be curious about the dynamics that I showed you in the diameter of those circles in the heat map that I showed you. So this shows you single-cell genomes, again, as the references, and metagenomes using to quantify the relative abundance of that population. So the number of reads mapping to each single-cell genome. And on the top, I have the AC1 Actinobacteria. On the bottom, I have those LD12 bacteria. And as you might expect, that because we can't separate their populations as well, they look to be much more synchronized through time compared to the AC1 Actinobacteria, which are, some of them are synchronized, some of them are not. And so you can start to think about which ones are ecologically distinct and responding to the same environmental variables or same kinds of interactions between other organisms and all those kinds of questions. And so these are some of our lingering questions that we're working on with Rex. So when do these sequence discrete populations exist, and when do they not? Do they ever not, or only for certain phylogenetic groups? And then I'm very interested in the lifestyle of the organisms and how that relates to their population structure. So is there something about how they interact with other organisms, how they respond to the environment that might be explaining some of those patterns? And then starting to get into the more mechanistic questions about what are the forces that make these organisms change in terms of the diffusion or diffusivity of their populations, if you will. And so I just wanted to circle back a little bit to what I talked about earlier with the power of time series concept. So if you apply this concept of the invisible present that John Magnuson developed for ice cover in Lake Mendota, or any kind of thing that you're looking at over many years to decades in ecosystems, that if we had only looked at Lake Mendota in the year 2006, we would have seen this. And we might have seen, okay, well, AC1A and B and C are abundant and they have these certain patterns, but they sort of taper off in the middle of the year 2006. If you expand that a little bit, you start to see more, and you think, oh, wow, there might be some annual patterns there. And then you can look at the whole time series and start to look for chunks, or at least phases that the lake has gone through. We can start now overlaying things that are happening to the lake. We know that there's been a zebra mussel invasion in the past few years. We had spiny water flea invasion. We have floods. We have trends in climate, so we can see El Nino cycles. And so all the things that we can see in other metrics that people care about in lakes, we can see in the bacterial populations, and think about how all those things are connected. So I guess my take-home message is that if you study natural ecosystems, you should be sampling them a lot. And I always tell the students in the room, ask your PI to buy you another -80 freezer to store all those samples so that you can go back and sequence them later when it becomes super cheap to sequence those samples. Because that's the only way that we've been able to do this, is that back in 1999, the person who I inherited this project from had the foresight to just start putting samples in the freezer. Now we can go back and sequence as many of them as we want. And so with that, I'll acknowledge my colleagues in crime. So I mentioned that I work a lot with the folks at Uppsala University, people here at JGI, especially Rex, Tanya, and Susanna. And Ramonis provided some of our early single-cell genomes, and then the folks in the Earth Microbiome project that contributed some stuff, my students, and the people that fund us. So thank you for listening. Go Freshwater.
Thank you, Trina. I'm sure we have some questions. In the strike. Yeah. Oh, sorry. Thanks, Sean.
Hi, thanks. So I had a question about, because in some of your plots there were definite dips down to really low relative abundance. So do you ever see, so you said you hate, not hate, but eukaryotes.
I can't handle eukaryotes.
But you must be getting eukaryotic reads.
Oh.
Are you? And then the question would be, do you see any evidence of like predator-prey relationships in some of these patterns that you're observing over time?
We actually haven't looked at the predator-prey aspect of it. For some of the dips in one of the plots I was showing, that's 16S primers, so that was just bacteria. For the metagenomes, we do see eukaryotic reads. We tend to just take them out, and we haven't looked at how they relate to the bacterial communities. They were much less abundant than we expected them to be. So they're probably like maybe 10% of the metagenomic reads, which surprised us because we don't pre-filter our samples. We just take whole water and put them on 0.2 micron filters. So we were worried that we were going to get 90% of the reads were going to be zooplankton, but they aren't. So we can focus on the chunk of reads that are bacterial. But that would be an interesting, really cool thing to look at. I would love to have a student or a postdoc take that on.
Hi. In your comparative analysis of the single genome of these bacteria, in the region where you see discontinuity, do you see a signature of the genes or the portion of the genome that's responsible or within that disparity or the changing region?
Like which gene? So yeah, it's interesting about which genes are recruiting and above 95% and then which ones are recruiting below 95%. What we found is that if you take two genomes, two single-cell genomes, and compare them to each other, either they all their reads, or sorry, all of their genome aligns at above 95%, or all of it aligns below 85%. So it's not as if there's different. There's a few exceptions, but there's not sections of the genome that are shared at a high identity and sections that are shared at a low identity. That tends not to happen. So that's why we're also pretty sure that if we slice it at 95%. Everything above 95% is part of the population that we're interested in, and only part of that population that we're interested in. So we've done a lot of pairwise comparisons of our reference genomes to really solidify that. And that's also in the Vargas paper from JGI too. So there are some exceptions, but they're pretty rare. More than we expected, for sure.
Are the lake bacterial genomes as small as genomes in oceanic bacteria?
Yes, they are. And they're so when we published the first genome from the AC1 Actinobacteria, our genome size estimates, it wasn't complete. We estimated that it was smaller than Pelagibacter, and we were all excited. We have the smallest free-living genome, haha. But Steve Giovanoni still didn't care. But people now have closed genomes, and they are a little bit bigger than Pelagibacter, so they're about 1.2, 1.3, but still much smaller than what you would expect, and certainly from cultured representatives, which seems to be the story now that's unfolding for both aquatic and, I think, terrestrial organisms.
So you looked change on bacterial population through time, but did you look in different locations? It's maybe different behavior, no? Different location of lake, maybe completely different, picture.
Yeah, so we only look at one location in Lake Mendota, so we have to make a decision about one point over many time points or multiple, so we don't do much spatial stuff in Lake Mendota, but we have looked at other lakes. Rex led a paper that came out of our data set from a bog lake in northern Wisconsin where we saw similar kinds of patterns, except that in one genome out of the 30 that we looked at very closely, we had a complete sweep of genetic variation. So basically a genome-wide sweep that purged all of the diversity in the genome. So it went from being, 98, 97% identical to being 100% identical in the population over a time span of about 5 years, also in that lake. It happened to be a chlorobe, so if anybody knows much about green sulfur bacteria, maybe there's something that explains that, but it was only one population, and so that one looked different from the others. Those are the only two lakes that we've done this in so far. Because you need to have the time series, and you need to convince JGI to sequence it for you. Then you need to convince the student to actually look at the data.
One last question.
So I was curious, you've talked about sampling across multiple timescales. What's the shortest timescales you've sampled across? I mean, do you see day-to-day variation in these samples?
We've done that. We did that before we started doing metagenomics. So we did it using 16S. And the conclusion we came to, at least for our temperate lakes, given the rate at which these ecosystems are spinning, if you will, is 2 or 3 times a week is the optimal to capture the dynamics that we see, at least with 16S. We can't do that logistically even. I mean, lakes are easier than oceans, but not that easy. So we usually do once, maybe twice a week, at least for Lake Mendota. Our lakes that are farther away, we often do once every other week. But we do have some data sets where we've spatially surveyed the lake in time as well. And so those are by sample, the sample matrices are huge, and we haven't done metagenomic sequencing on those. So I'm really interested to compare the extent of temporal variation to the extent of spatial variation, because Lake Mendota is big enough that you get patches. It's, again, not as heterogeneous as the ocean, but is it more different from one end of the lake to the other versus like from today till tomorrow at one location? So those are really interesting questions too.
Do you see diurnal variation as well?
In gene expression, not so much in bacterial community composition. I mean, cyanobacteria are different, right? Because they can migrate, they can physically migrate. So depending on how you collect your sample, you see them or you don't. But the heterotrophs, not really.
Okay. Thanks a lot, Trina.
Thanks.